Okay, I didn't even change a slide. I meant to. All right, chapter 14. This is our non-specific immunity, our first and second line of defense. So our first line of defense is referring to the physical and chemical barriers, like literally your skin is like the biggest one, right? That keeps things from getting in. They're pretty much that simple. The second line of defense is talking about the cells that are involved in innate immunity. Innate means non-specific immunity, but the cells that are made for immunity. Whereas like your skin obviously isn't just for immunity. So the second line of defense is referring to your actual immune cells, but they are sort of going to have a non-specific response to things. What's the difference? Well, when we talk about the third line of defense, that's going to talk about your T cells and your B cells and antibodies and all that, but those are going to respond very specifically to specific bad guys, very specific bad guys, or specific parts of the bad guys even. So that's what we're going to get into the next one. This one's the general stuff. These guys are the ones that are going to see the bad stuff right up front and just start reacting because they know it's bad, but they don't care really specifically what it is. So they're going to start mounting a response as soon as they can to try to keep things under control, while the specific responses are building up and preparing to go to war because they haven't got their stuff together yet. They have to know which soldiers to send out first. So our first line of defense, the physical and chemical barriers. Lysozyme is an enzyme that is produced by a lot of different parts of your body and actually is in your phagocytic cells, which are a big part of your innate immunity. It's an enzyme that breaks down peptidoglycan. And remember, peptidoglycan is part of the bacterial cell wall. Keratin is a waterproofing protein that sort of seals the skin so that it's harder for things to get inside and so that we're waterproof and all of that. So that's kind of part of the first line of defense. Anything on that outside, things like no nasal hairs and all that, that's part of that first line of defense. So more important for this chapter, I feel like, is the second line of defense. So that's our part of our immune system, specifically our non-specific immune system. First, we have to be able to distinguish between self and non-self. So what is the bad guy and what is our self? We don't want to just attack anything that interacts with us because sometimes those proteins are going to be associated with broken down self cells. And we don't want to just start attacking our self cells because that's where autoimmunity comes from. So that's what auto means, by the way, in case you didn't know that. For those of you who didn't know that, autoimmunity is when your body attacks yourself. Auto is being yourself. So we want to avoid that. So the way that we distinguish self versus non-self, we have certain markers on the surface of our self. These are proteins, glycoproteins, sugars associated with the protein, or whatever on the surface of a cell that we can recognize as self. We have, as we've developed in the womb and as we've grown up, been exposed to these molecules and hopefully been taught that these are safe molecules to be seeing. That's what those are. So we've trained ourselves to see those as self. The lymph system itself is going to include the thymus, which is about here. The lymph nodes all throughout your body. You already knew that. You don't just have them in your neck. They're in your entire body. And most of them, no diagram is true to this ever because it's just hard to draw it all. But most of your lymph nodes are going to be in your gut, actually. That's the highest number of your lymph nodes. A lot of them in your gut. And that's because of all those bacteria and exposure to bacteria that you have going on down there. A lot of your training to determine which bacteria are friendly versus non-friendly happens in your gut. So you need the lymph nodes there to keep everything closed so it functions properly. The spleen, which I bet you guys barely know what a spleen does. Almost nobody does. So the spleen, yes, it does filter out damaged red blood cells. It's very important for that, actually. Don't underestimate how important it is for that. But it's also part of your lymph system and it works as a lymphatic tissue. Then we have the lymphatic fluid itself, which is plasma, essentially, and sort of like the liquid portion of the blood. And yeah, it carries the white blood cells around the body. The way that it all works is the heart pumps everything out as part of the blood. It's all mixed together, right? Pumps everything out to the extremities, just like it normally does, mixed in with the lymph. And then everything will come back to the heart except the lymph. The lymph has to get pushed out of the tissue by mechanical motion. So things like walking and moving around actually help cycle the lymph back up and into the circulatory system. And so if you are just lying there after your surgery, your lymphatic system is just lying there also. And that means you're not going to heal properly and you're more prone to infection. And that's why they'll put those wraps on the legs that kind of like inflate and deflate to try to squeeze the legs to try to get the lymph moving. That's what that's about. People say, no, it's to prevent edema. Yeah, what do you think the edema is caused by, though? It's caused by this lymph settling in the tissues, in the extremities. So that's what's up. Okay, white blood cells. The big guys, white blood cells are your immune cells, basically. Let's not forget that. White blood cells, immune cells, yes. These are called leukocytes overall. And we have the granulocytes, which includes neutrophils. Anything phil is going to be your granulocyte. So neutrophils are the most common ones. Eosinophils are typically associated with allergy. And then we have the basophils. Then we have, right, well, they have all the phagocytes and all of that. But before I get into that, the other kinds of leukocytes, other than those granulocytes with the phils, we also have the agranulocytes, and that includes monocytes. Monocytes, when they migrate out of the bloodstream and into a tissue, differentiate into macrophages. And one kind of monocyte that differentiates is going to be, and gets into the skin area, is specialized kind of macrophage known as a dendritic cell. So those are kind of in the skin tissue area. And then we have the lymphocytes. The lymphocytes are your T and B cells, and those are involved in your specific immunity, targeting specific pathogens. And those are the ones that are going to carry the memory to fight against specific things. That's what we want to target with our vaccines. So going back to those granulocytes, neutrophils, monocytes that we were talking about with the agranulocytes and the macrophages, there's these mast cells. Those are often involved in allergy as well as Hellman's infections. And then the dendritic cells, all of those guys are phagocytes. And some of the phagocytes work as antigen presenters. They'll essentially take whatever they eat up. Phago means eat, by the way. So eating cells. So they'll go out and they'll eat whatever up, and they will break it down and then decide this, is something that I'm interested in. This doesn't look right. So we're going to pop it out on the surface of our cells and go find somebody to confirm that this is bad stuff. This is related to a bad guy. Those cells that do that and interact with T helper cells that are going to instigate, everything going on later, we'll talk about in the next chapter. Those are called antigen presenting cells. So they're the ones that typically are going to be bringing that up to the T cells. All right. PAMPs, P-A-M-P. These are pathogen-associated molecular patterns. This includes things like lipopolysaccharide or endotoxin. So they will automatically see those and automatically say, bad guy, bad guy, bad guy, bad guy, we need to start fighting. So stuff like that is non-specific response, because a lot of gram-negative bacteria have lipopolysaccharide, right? Gram-negative bacteria that have that, we're going to start calling it a bad guy when we see it. So that's just one of the examples. I don't know. Phagocytosis, there are steps involved. These are the steps that's listing here. So chemotaxis, that means it was signaled to an area maybe by inflammatory cytokines. A cytokine is like a call to arms chemical. So we get called to the area, chemotaxis, taxi, like moving, reacting to some sort of chemical signal. Adhesion, okay, we've set up shop in wherever this is going on. We will, and we are going to adhere to whatever the bad guy is in that area, or the bad guy will adhere to us. And then we're going to engulf it. So literally just stretch our membrane around it like a big old bite. and then the formation of the phagosome. So that is, we engulfed it, and then we took that vesicle inside of our cell. Well, now we're going to try to fuse it with a lysosome. A lysosome contains things like lysozyme that breaks down peptidoglycan, as well as other molecules that should be able to help with killing bad guys, bad pathogens. And then that lysosome that contains those bad chemicals, killing chemicals, will fuse with the phagosome that we ate the bacteria up into and then kill the bacteria. So that's the destruction killing part. And then we're going to release whatever debris that we made from that interaction. So that's the idea of how phagocytosis essentially works. The inflammatory response, we brought this up a little bit in the last chapter, but here's some of the terms associated with it. Rubor is the redness, calor is the warmth, tumor is the swelling, and dolor is the pain. These are just vestiges of the good old days, man. Some terms referring to stuff, but the inflammatory response is important. We do have to have releasing of chemokines and cytokines, our call to action molecules, and start having inflammation, because this is our first exposure to whatever BS just came into our body. We need to start fighting it now, because it's going to take a while before our specialists come in. So our frontline guys need to start the fight first. So I don't know, these are the Marines. So fever, any pyrogen can start a fever. So that's what pyrogen means. It's any molecule that can elicit a fever response. Fever increases metabolic activity, decreases microbial growth, releases transferrin to keep iron away from the microbes, and it can be caused by exogenous sources, so things like lipopolysaccharide that came from outside of our body, or other things from outside of our body from the pathogen, can automatically trigger fever production, or very commonly will have endogenous signaling. And remember, so endogenous signaling is going to be inside of signals from inside of our cells. So those call to action molecules, we have production of certain ones that will be, activated via certain pathways in response to certain infections. As those get triggered, some of those are going to be pyrogens and cause development of a fever, which means that the body thinks that you should be responding with a fever to this infection. All right, so the endotoxin, that is LPS, so lipopolysaccharide. That's the PAMP that is associated with gram-negative bacteria. Interferon is one of these, I keep saying cytokines and stuff. That is one of those molecules. Interferon is. It's just a call to action molecule. Now, this one's special, not particularly, but it's one that we're going to be talking about anyways. It's only special because we're talking about it. Interferon is an innate defense against viral infections or any bad thing going on within the cell, including cancer. So the virus gets into the cell. The cell is trying to fight off the virus, and it's probably not going to win as the virus is being made. However, parts of the cell has recognized that this virus is a bad thing that's going on in its cell, right? And something has interacted with those viruses and triggered production of interferon. So the viral infection in cell A triggered production of interferon molecule, and the interferon is released by cell A, and now cell B next to it gets that message. It's like a secret message sent to it, and it reads the message and it says, "We got some viruses coming, buddy, ready up." And then it does, it readies up. It starts making up antiviral molecules and getting ready to fight off viral infection, so that once the virus shows up at its doorstep, it has a shotgun ready. So that's basically the idea with that. Then we talk about complement cascade. I really like the complement cascade. Complement is so useful in immunity. We can trigger it through non-specific immunity, or we can have it be triggered by a specific trigger. So the classical complement pathway is we're triggering it through antibodies. And that would be the specific trigger, specific response trigger. So basically antibody binds onto a bad guy, complement proteins come and interact with that. And there's this whole cleaving. And then if this is cleaved, then we're going to produce this. And then that's going to be cleaved and it binds with this. And that's the complement cascade. That's why cascade. They all interact with one another. And eventually we'll get down the line to the following thing. We can lead to inflammation. So some of the products that are produced in the cascade will cause the inflammatory response, which is, like you're starting to probably learn, incredibly important and very common. It can cause opsonization, which is a new word for you guys. Opsonization is whenever we are putting molecules on the outside of a bad guy cell to make it easier for our phagocytes to eat them up. So it helps phagocytosis, basically, helps eating up the bad cells. Like if it has a capsule, it's hard to, like we already said, capsules make it hard to eat bacteria. So opsonization can help with that. Cytolysis is probably the coolest thing the complement cascade allows us to do. And that is, it has, as we're going through the cascade, we will trigger all of these little proteins to be made that can form a hole, a little like tunnel through the membrane of the pathogen. And then the insides of the pathogen can leak out and then cause it to die. Or other bad molecules can get in to kill it. I think that is so cool. You guys should think that's cool that your body can do that. And this is just your innate immune response, just your first response. Man, I can't wait to get into the next chapter with you guys and talk about specific immunity with the B cells and the T cells. It's a lot if you have never heard of this before, but it is so cool. And yeah, so I'll see you guys next time.